Hello, everyone. My name is Allison, and I'm here with the Bioprinter sub-team for Professor Steven Santana's CUTE Lab at Harvey Mudd College in Claremont, California. Over the years, 3D printing has become a popular technology and emerged as a potential solution for cell communication, drug delivery, and tissue engineering. Its ability to produce complex biomimetic structures and compatibility with handling fragile biomaterials has furthered benchtop research and development in healthcare.
We are redesigning a consumer FDM 3D printer to create a benchtop model 3D bioprinter capable of simultaneously extruding multiple bioinks to produce biomimetic structures. The printer utilizes a pressure-driven system and extrudes bioink into fresh or free-form reversible embedding of suspended hydrogels. Fresh is made using nanoprecipitation of gelatin pluronic solution in IPA, and it allows for suspension and constrains the motion of bioink while allowing free motion of the extrusion needle, which facilitates printing complex geometries and overhangs.
Our bioprinter, which is depicted on the right, consists of a heavily modified Creality Ender 5 Pro 3D printer with a Duet 3 6HC controller board running RepRap firmware, a PID pressure pump system based on the micropump project, and a Raspberry Pi to control everything. In order to get the bioprinter to print an actual file, we devised the following system, which corresponds to the flowchart on the left. First, the object to be 3D printed is added to Prusa Slicer, a program that converts a 3D model into a series of line-by-line instructions that would tell a conventional FDM 3D printer what exactly to do. These instructions are in a format called G-code. Next, the slicer calls a custom Python script to post-process the G-code into a form understandable by the bioprinter. The main thing that the script does is use the movements presented in the G-code file to predict the pneumatic pressure required to achieve a desired bioink flow rate at any given moment in time. This pressure is then added as a custom auxiliary command to the G-code file. Lastly, the G-code file is run on the printer, which then requests a specific pressure from the pressure pump. This pneumatic pressure displaces bioink from a reservoir on the printhead, which is a bit difficult to see in this picture, extruding it through a nozzle into the fresh gel. One of our team members, Carlos, will further expand on the pressure pump system.
Currently, we have a pressure pump system which we developed during the spring semester, which is able to control pressure based on a desired pressure we give. Basically, the system would first start giving a pressure of how much it desires to have, and then it would then output the pressure. It would then check how much pressure we actually have and then adjust for any difference in pressure. This is pretty useful since it's a pretty good way to control precisely the pressure. And other than these slight modifications that we would do on using 3 channels of the 8 that the pressure pump was designed for, we changed the communication to be mainly through UART and not the GUI that was provided by the research paper we found this in, which allows us to communicate more directly with the Raspberry Pi. Another small thing we have changed is the printhead, which now allows to have a pretty well holding system for different sizes of needles, which is pretty convenient if we want to change those variables when we're testing different printing. Currently, we have only done some amount of testing with sodium alginate, which we can see here in the graph on the left. We have some 4.1% sodium alginate shear reality stiffness ranges we have calculated and measured. In this case, we can see that the range is pretty wide, which is not ideal. And we're currently going to be working in trying to make this smaller for future work. The current system also is being modified and adapted to use different kinds of bioinks like polyethylene glycol, which in this case would allow us to have a system that polymerizes using UV light instead of calcium chloride, which we will explain pretty soon. Allison will explain why this is pretty convenient for us.
Yeah, so by using Freshgel as the port path for the suspended bioinks, we're able to maintain the shape of our desired prints. The image on the right shows 3 different tests we recorded when we were using the modified printer and the G-code that we created. So the first test was done without any curing and was printed in air. It's only able to hold any 2-dimensional prints. Meanwhile, without any polymerization, such as using calcium chloride, which we use with the sodium alginate, but without it, the bioink diffuses into the fresh gel and it just completely loses all its structure. Only when printing in fresh gel and polymerizing the extruded material afterwards with the calcium chloride were we able to create this sound structure. While some food coloring is lost over time, the main goal of our research, which we were able to achieve, was creating these complex geometric shapes via the hydrogel printing.